Namaste. Myself Dr. Riva Gupta from JAYOTI VIDYAPEETH Women's University, Jaipur, Associate Professor in Department of Kriyashari, Faculty of Ayurveda Science. आज का हमारा सेशन है सेल मेम्ब्रेन फिजियोलॉजी फॉर बीएमएस फर्स्ट ईयर स्टूडेंट्स. Cell membrane, synonyms plasma lemma or plasma membrane. It is a protective sheet enveloping the cell body. It separates ECF and ICF. It is semi-permeable membrane. Its thickness is about 75 to 111 Armstrong. Composition 75% proteins, 40% lipids, and 5% carbohydrates. Structure. Its structure is fluid mosaic model. In this, membrane is a fluid with mosaic of proteins. Mosaic means pattern formed by arrangement of different colored pieces of stones, tiles, or glass, etc. So in this model, mosaic are proteins. According to this, in cell membrane, proteins are found to float in the lipid layer. Lipid layer of cell membrane. It is bilayered structure and formed by a thin film of lipids. Lipid layer is not a solid structure. It is fluid in nature. The major lipids are phospholipids and cholesterol. Functions of lipid layer. It forms a semi-permeable membrane. It allows only the fat-soluble substances like oxygen, carbon dioxide, and alcohol. Protein layer of cell membrane. These layers cover the two surfaces of central lipid layer. Two types of proteins, integral proteins or peripheral proteins. Integral proteins pass through the entire thickness of the cell membrane from one side to another side. For example, cell adhesion proteins, cell junction proteins, carrier proteins, channel proteins, hormone receptors, enzymes, and antigens. Next is peripheral proteins, which are partially embedded in the outer and inner surfaces of the cell membrane and do not penetrate the cell membrane. For example, proteins of cytoskeleton, some carrier proteins, and some enzymes. तो इस डायग्राम में हम देखते हैं कि लिपिड लेयर है और प्रोटीन लेयर है. लिपिड लेयर में जो मॉलिक्यूल्स हैं ये पिन शेप्ड मॉलिक्यूल्स होते हैं जिसमें एक हेड होता है और एक टेल होती है. तो जो हेड होता है वो बाहर की तरफ मीन्स जो सेल मेम्ब्रेन होता है सेल के बाहर की तरफ रहता है और जो क्योंकि मतलब बाहर और अंदर की साइड में हेड वाला पार्ट होगा जो कि हाइड्रोफिलिक होता है और जो टेल वाला पार्ट होता है वो हाइड्रोफोबिक होता है तो वो बीच में होता है. एंड प्रोटीन्स होते हैं यहाँ पे हम देखते हैं जो ग्रीन कलर में दिखता है कि कुछ कार्बोहाइड्रेट मॉलिक्यूल्स है तो कुछ मॉलिक्यूल्स प्रोटीन से जुड़े हैं तो हमने उनको ग्लाइकोप्रोटीन बोला है और कुछ मॉलिक्यूल्स लिपिड से जुड़े हैं तो फिर हमने उनको ग्लाइकोलिपिड्स बोला है. मीन्स ऐसे प्रोटीन्स जिन पे कार्बोहाइड्रेट जुड़ा है उन्हें ग्लाइकोप्रोटीन कहेंगे और ऐसे लिपिड्स जिन पे कार्बोहाइड्रेट अटैच है उनको ग्लाइकोलिपिड कहेंगे. देन सम there are some cholesterol molecules also. Then outward facing layer of phospholipids and inward facing layer of phospholipids. Integral proteins, peripheral proteins, filaments of cytoskeleton. Polar head of जो head है polar head of phospholipid molecules. Then non-polar tails of phospholipid molecules and bimolecular lipid layer contains protein. ये हम देख सकते हैं कि जो मेम्ब्रेन है वो सेलेक्टिव परमेबल होती है तो जो स्मॉल अनचार्ज मॉलिक्यूल्स लाइक H2O, अमोनिया, CO2, ऑक्सीजन ये इसमें से पास आउट हो सकते हैं. प्लस जो लिपिड सॉल्युबल सब्सटेंस है लाइक हाइड्रोकार्बन है, अल्कोहल है ये इसमें से पास आउट हो सकते हैं. बट जो वाटर सॉल्युबल सब्सटेंस लाइक ग्लूकोज है ये इसमें से नहीं निकल सकते हैं. एंड आयंस है लाइक सोडियम, हाइड्रोजन एंड क्लोराइड आयन, कैल्शियम आयन ये भी आयंस इसमें से नॉर्मल कंडीशंस में आयन पास नहीं हो सकते, ट्रांसपोर्ट इनका नहीं होता. ना ये आयन सेल से बाहर से अंदर आ सकते हैं ना अंदर से बाहर जा सकते हैं. फंक्शन. Integral proteins provide structural integrity. Channel proteins act as channel for diffusion of water-soluble substances like glucose and electrolytes. Carrier proteins transport substances across the cell membrane by means of active or passive transport, or act as a pump by which ions are transported actively across the cell membrane. Receptor proteins serve as receptor sites for hormones and neurotransmitters. Enzymes control the chemical reaction within the cell. Antigens induce the process of antibody formation. Cell adhesion proteins are responsible for attachment of cells to their neighbor cells. Then carbohydrate of the cell membrane. There are some carbohydrates which are attached to either the protein or the lipid, which are called glycoproteins or glycolipids. All these carbohydrate molecules form a thin, loose covering over the entire surface of the cell membrane called glycocalyx. Functions. These are negatively charged and do not permit the negatively charged substances to move in or out of the cell. Help in tight fixation of cells with one another. Work as hormone receptors. Functions of cell membrane. Protective function for cytoplasm and cell organelles. Act as semi-permeable membrane, which allows only some substances to pass through it and act as a barrier for another substances. Nutrients absorption. Next is excretion of metabolites and other waste products, exchange of gases, and maintenance of shape and size of the cell. Next is transport through cell membrane. Transport through cell membrane is necessary to get nutrients, electrolytes, water, etc., and get rid of many unwanted and waste substances. The lipid and proteins of cell membrane play important role in the transport of various substances from ECF and ICF. There are two types of basic mechanism. First is passive transport and another is active transport. Passive transport, it is also called diffusion or downhill movement because transport along concentration gradient or electrical gradient or electrochemical gradient. It doesn't need any energy. It is of two types, simple diffusion and facilitated diffusion or carrier-mediated diffusion. Then simple diffusion is of two types, through lipid layer or through protein layer. First is simple diffusion through lipid layer. Only lipid-soluble substances like O2, CO2 and alcohol. More solubility, more diffusion. Simple diffusion through protein layer for only water-soluble substances like electrolytes. It is conducted by protein channels or ion channels which are selective permeable. Means one channel for one ion. Some are continuously open, means ungated channels, and some are closed, means gated channels, which are open when required. The gated channels are of three types: voltage gated, ligand gated, and mechanically gated. Voltage gated, which opens when alteration in voltage; ligand gated, which opens when chemical changes; and mechanical gated, which opens when there is some change in pressure, temperature, etc. Then facilitated diffusion, it is faster process. Water-soluble substances having large molecules like glucose and amino acids. It transported through cell membrane with the help of carrier protein. Then special types of diffusion, bulk flow, filtration, or osmosis. First is bulk flow. Bulk flow means diffusion of large quantity of substances from a region of high pressure to the region of lower pressure. It occurs due to pressure gradient across the cell membrane. Importance: exchange of gases across respiratory membrane in lungs. Due to partial pressure of difference between partial pressure of oxygen in alveolar layer and alveolar capillary blood, and partial pressure of carbon dioxide in alveolar air and alveolar capillary blood. Next is filtration. Movement of water and solutes from an area of high hydrostatic pressure to low hydrostatic pressure. Hydrostatic pressure developed by weight of fluid. Importance: formation of tissue fluid, formation to tissue fluid, glomerular filtration in kidney. Osmosis: movement of water and other solvents from an area of lower concentration to an area of higher concentration of solute through a semi-permeable membrane. In this diagram, we can see that there is lower concentration of solute or sugar, and one side there is higher concentration of sugar. From semi-selectively permeable membrane or semi-permeable membrane, H2O means water goes from water, which is solvent here. So then this solvent moves from lower concentration to higher concentration of solute. Semi-permeable membranes permits the passage of only water or other solvents, not solutes. Endosmosis, movement of water in the cell, and exosmosis, movement of water out of the cell. Importance: absorption from intestine, regulation of urine formation, and exchange between plasma and red blood cells. Next is active transport. Transport of substances against chemical or electrical or electrochemical gradient. It is also called uphill transport. It needs energy which gains from breakdown of ATP. Transport by carrier protein across the cell membrane. Carrier protein transporting only one substance are called uniport or uniport pump, and carrier proteins transporting more than one substances are called symport and or antiport pump. Symport, when two molecules goes in same direction, and antiport, when two molecules goes in opposite direction. Mechanism: substances come near the cell, then combine with carrier protein, which form substance protein complex. Then substance protein complex move towards the inner surface of cell membrane, where released from carrier protein and where substance released from carrier protein and carrier protein moves back to the outer surface of the cell membrane. Active transport is of two types, primary and secondary. Primary active transport, mechanism in which energy is liberated directly from the breakdown of ATP, like transport of sodium, potassium, calcium, hydrogen ion, or chloride ion. Secondary active transport, transport of a substance with sodium ion by a common carrier protein, either in same direction or in opposite direction. Energy released by the movement of sodium is utilized for the movement of other substances. It is of two types, co-transport and counter-transport. Carrier protein that transport two different molecules in the same direction across the cell membrane is called symport and or symport pump, and this type of transport is called co-transport. And that transport in opposite direction is called antiport or antiport pump, and this type of transport is called counter transport. So now here we can see sodium potassium pump. Sodium goes from outside to inside, energy liberated in form of ATP, and potassium ion comes out inside from outside. And here are some examples. Sodium and calcium exchanger and opposite sides, sodium and H plus ion exchanger, chloride and bicarbonate ion exchanger, and calcium and sodium ion exchanger. Then sodium cotransport. Another substance is transported along with sodium by symport, like glucose, amino acid, chloride, iodine, iron, and urate. Sodium counter transport. Another substance is transported across the cell membrane in exchange for sodium ion by antipode, like calcium, hydrogen, and magnesium. Next, some special types of active transport are vesicular transport, endocytosis, exocytosis, or transcytosis. Endocytosis, transport mechanism by which the macromolecules enter the cell. It is of three types: pinocytosis or cell drinking. It is for small molecules by which bacteria and antigens are taken into the cells. Phagocytosis or cell eating, through which larger bacteria, larger antigens, and other large foreign bodies are taken inside the cell. Only few cells like neutrophils, monocytes, and tissue macrophages show phagocytosis. Next is receptor-mediated endocytosis, transport of molecules with the help of a receptor protein present on the surface of cell membrane. For example, transport of hormones, lipids, toxin, bacteria, antibodies, etc. Next is enzocytosis, process by which substances are expelled from cell without passing through the cell membrane. Next is transcytosis or vesicle trafficking or cytopempsis, process in which an extracellular macromolecule enters through one side of cell, migrates across cytoplasm of the cell, and exits through the other side with the help of receptor protein. Example, transport of HIV pathogen. Now, here we can see process of endocytosis by phagosome or vesicle, pinocytosis, phagocytosis, pinocytosis, or receptor-mediated endocytosis, and another for exocytosis. There are some questions, and here some reference books. Thank you.